Our protocol allows bacterial dissemination and burden tracking in an experimental neonatal sepsis model in real time, and the ability to correlate other markers of disease with the pathogen burden.
This protocol allows us to longitudinally analyze sepsis in individual neonatal mice, providing the opportunity to observe and compare infection dynamics and bacterial dissemination in real time.
Preclinical testing of interventions for neonatal sepsis can be performed with this method, allowing for the monitoring of the effects of host control of the bacteria.
Begin by placing age-matched pups into either high or low-dose litter groups within a biosafety level 2 cabinet. On postnatal day 3 or 4, record the weights of all the pups prior to inoculation. Load insulin syringes with PBS, or the high or low-dose E. coli Lux inoculum in the biosafety cabinet, and place the loaded syringes on ice. Place the neonate to be injected onto a clean surface within the biosafety cabinet, and raise the skin at the nape of the neck as if to scruff the pup. Insert the needle bevel up just beneath the skin in the space created between the skin and the muscle. When the needle can be felt under the skin, inject 50 microliters of the inoculant while simultaneously releasing the pinched portion of the skin to prevent backflow. Then, remove the needle slowly and with care. When all of the pups have been injected in the same manner, return the pups to their dams. Immediately after